as it take us through how to write a Mikhail workshop paper. Over to you, Professor Mehmet.
Yes, so let's see. Let me try share my screen. I might have to switch back because I'm going to also show an example paper. Hi, everyone. Thank you, Mario, for the invitation and for the introduction. So I'm just going to briefly go over. I would love to keep this as a Q&A interactive. So I'll just briefly go over like the basics of how to write a Mirasol Mikai workshop paper. Can you see my screen?
Yes, we can see
You
it.
can, right? Okay, cool. Awesome. So, I'll just start with the submission guidelines first. These are just, you can find this on the Mirasol Workshop website as well, but I'm just going to go over them. And in case you have any questions, please let me know. So, submissions are invited as long papers for this workshop. So, there will be 8 pages of contents plus 2 pages for references. So, this is a strict requirement by Nikai. So, please follow the guidelines. You can use LaTeX or Word. And I'm gonna go over the templates in a little bit, but you can use either of the format. It's totally up to you. However, the final submission needs to be in PDF, just so you know. The review process will be double blind, meaning the reviewers will not know your institution or your names, or you will not know the reviewers' names. So this will be a double blind process. Therefore, please anonymize your submissions to prevent identification, meaning do not include during the review process, for example, links to GitHub pages, right? Because that's a direct identifier. For example, that's one of the most common problems that we identify. Or do not say things like, in our previous work, and put a reference, we did this. No, don't do that. All right. And each submission will be rigorously reviewed by at least 2 external reviewers to assess suitability to the workshop, Mirasol workshop. Um, in terms of publication, accepted long papers will be included in the workshop proceedings, which will be published. So these are citable papers. So it's really good for your careers. So we're really excited to get those submissions in time. And accepted submissions, so accepted papers, after you get the acceptance, you can definitely post the preprints on archive. That's totally fine, and we encourage that. But you cannot post the camera ready version. Meaning, after we accept your paper, we will work with you to make sure that your paper fits all the guidelines by Mikai, and that's what we call the camera ready version. That's like the preceding version of your paper. You cannot publish that during the publication embargo period. All right. So the paper templates you can access all of these links through the Mirasol Workshop website. I include the QR code here, so you can find both the Word and LaTeX versions of the paper template on our websites, and you can also find it under How to Submit tab of our website, and you will see that there's a unique LaTeX template and Word template. So, uh, if you have any questions on that, please let me know. All right. Now, suggested paper structure. This is just suggested. It's not required, but we do strongly encourage you to follow something that looks similar to this. And I'm going to give you an example from one of my groups, one of my graduate students from a paper from last year. So you start with an introduction and background. What does this mean? You can, for example, talk about the health problem that you're studying here. Like, let's say you're studying breast cancer. And you could talk about the importance of breast cancer and what's the unsolved problem that you're trying to solve. And this could be the introduction and the motivation for your work, essentially. Previous work can be, let's say you're doing lesion segmentation for breast cancer, right? What have people done in that domain, right? What sort of model architectures they followed and were they successful, right? That can be the previous work. Methodology is your essentially methods. You don't mention results here. This is you specify the model architecture that you work with. How did you split the training validation and cross validation and test data sets? What's your inference time? So we do encourage you to talk about computational resources and inference time in methods, primarily because this is the main motivation, right? We're essentially focused on resource limited settings here, so we do want to know what kind of computational expense you had in terms of running these models, right? So that's methodology, and the results. strictly talk about results. Like let's say, again, going back to the breast cancer example, let's say you are dealing with segmentation tasks and you want to report the DICE scores and HD95 scores. And you strictly, the format that I like to follow is results is you can think about it as you're talking about results with any commentary, without any commentary or emotion into it. So you just state facts. These are the results that we have. This is what we observed, right? Our model achieved this score and our baseline model for comparison achieved this score. So it's just statement of facts. You can think about this section that way. The discussion is now you're interpreting the results. You're talking about why, for example, your model overperformed the baseline model, you think? Or what does this mean clinically, like the improvement that you get? Or why does your model not uh, perform as you expected? So this is where you talk about essentially implications and interpretation of your results, and that's section 5. And a required section that we added this year is impact and resource constraint settings. So we do want you to talk about what's the impact of your work for resource constraint settings. And that's the last section. And this is a new section that has been added. All right. Um, Important dates. So full paper submission deadline is July 15th, and the camera ready will be September 3rd. These are strict deadlines imposed by MICAI, so there's no flexibility here. However, Maruf, correct me if I'm wrong. The travel, if you want to be considered for a travel award, I believe the deadline for submission is June 15th, right? Is that accurate, Maruf? I
Yeah,
don't have
that's
Maruf
correct.
confirmed.
That's correct. Yeah.
June 15th. Yes. But if you don't want to be, if you don't kind of want to be considered for a travel award, if that's not necessary, you can submit until you have until July 15th. All right. And this is an example paper. I'll stop here for like a minute for you to just take pictures and go to this link. This is a paper from one of my grad students Juan Pablo, who essentially submitted a workshop paper last year. So let me stop sharing and um, reshare my screen and just go over his paper just as an example. Here we go. So again, you don't have to follow this strictly this format. Do what makes sense for your paper. That's why I want to show this to you so that you can have an idea. Can you see my screen? Okay, cool. So this was a problem, essentially. This was a paper focusing on stroke segmentation and resource limited settings. So as you see here, you start with an abstract. This is essentially the summary of your work, right? Try to keep this concise. I would recommend 200 words at most. Um, and then in introduction, as you see here, Juan Pablo talked about why we care about stroke and stroke diagnosis and why that's important and how is it diagnosed right now. And why is it a problem in low resource settings? Like for example, in this case, um, in low resource settings, you typically don't have access to MRI. And because of this, right, you have to do ideally strokes diagnosis with non-contrast CT. And essentially, his paper is talking about this portion. Now, the second, instead of talking about um, uh, prior work in the second section, he actually talked about just data sets, essentially the data sets that have been released in this field. As I said, that's flexible. Here you can also talk about previous work in the second section, like what has been done in this field. I think kind of he talked about a little bit of that in the introduction, and then in second section he just talked about the data sets that are available. That is kind of like previous work, but not really. So as I said, it's flexible. And methodology is this third section. So as you see here, he talked about, for example, section by section. So you can have subsections. 3.1, you have how you essentially do skull stripping for these data sets. How you do vessel segmentation in CT and geography, that's his section 2. As you see here, he even put like an example of how this segmentation has been done. Our method, so we were doing intensity windowing for this method. So essentially, that's another step. And the model architecture, we use NNUnet for this. So as you see here, he talks about how we optimize hyperparameters, methodology. Then results, as you see here, we have the results of a baseline model and our model. We're comparing dice, and we're just talking about the fivefold cross-validation experiments, as well as this was a challenge paper called IELTS 2024. So we also talk about essentially our results and how kind of we overperformed in this case. The fifth section, which is, he called it analysis. As I said, you can call analysis, discussion, very flexible. The interpretation of the results. For example, here, he talked about why he hypothesizes that the model overperformed the baseline model. In this case, it was because we were doing this pre-processing pipeline of intensity windowing, which essentially enables the model to really focus on regions of interest. And that's what he talked about in this section. In conclusions, which is you can think about this conclusion section as like impact and resource constraint settings, which is again a new requirement this year. He talked about how this model can be useful for resource limited settings. That's about it. I'm going to stop right there. And if you have any questions, again, I'm more than happy to answer during the Q&A session. Thank you.
Thank you so much, Professor Memort. This is really excellent. One thing you didn't mention, which I will mention, is that that paper you presented was actually the winning paper for that particular challenge, and it is interesting to see
Uh,
that you are
Thank
giving
you.
it to us as a template for us to also present our own winning challenge in Mika as well. In the interest of time, I know we have multiple questions in the chat box here. I don't know if you quickly like to take them or do you like to take everything after we have the whole presentation? What works
I
best for
just
you?
sent everyone the paper link. So people are asking. So if you look at the chat box, is the paper link is right there.
Okay. So we have a question from Tony Foliage. I will be allowing him to quickly ask his question. Go ahead, Tony.
Hello, hello.
Yeah, we can hear you. Go ahead.
Sorry, it was a mistake. Yeah, no,
All right.
I'm sorry.
All right. So we also have another question from Samuel Ajayi, who's asking for a clarification between to be considered for the travel guard. Is the early submission July 1st or June 15th? We have an initial date of July 15th, but if you have a peculiar challenge, if you have a peculiar situation, you can send an email and we can see how we can accommodate this. But I wouldn't really advise that you delay till early July before you make your own submission. All right, so without further ado, I would like to invite Professor Natasha Lepo, who will be also taking us about the LISA challenge and how we can participate despite the fact that we are from resource constraint settings. The goal of the challenge was actually to synthesize diagnostic information from low field MRI images. Professor Natasha, over to you.
Yeah, hi everyone. Thank you for coming to the presentation. Really happy to be presenting the LISA challenge to all of you. Here I'll share my slides. Wait 1 second. All right, share. Here we are. All right, can everyone see my slides?
Yes, yes.
Okay, perfect. Let's see, full screen. Oh, give me a second. Sorry. How do I make it full screen? Right. Sorry, I should know this. I've done this many times before. Is it not full screen now?
Not yet full
Full
screen.
screen. There we go. Okay, I'll just leave it like this if that's okay with you
Thank you.
guys. Right. So the low field pediatric brain and magnetic resonance image segmentation and quality assurance challenge is now a multi-year challenge at MICAI. And our goal is to democratize just and get more input from the community for designing tools for ultra low field MRI. And this is based on a project that we are part of called the Unity Project that's funded by the Gates Foundation. And the idea of this project is to improve global neuroimaging access worldwide. And the way that this was done was by initially putting MRI, low field MRI scanners, and I'll talk a little bit about those in a minute, but putting those all over different countries around the world. I'll show you a map of where all these scanners are, but essentially then the idea is to look at environmental determinants of health in children 0 to 5 years old, using MRI and brain MRI more specifically, trying to see how children's brains develop, and how to optimize any environmental factors to make sure that every child thrives. And so this is what the ultra low field scanner looks like. For those of you who haven't seen one before, this is the hyperfine scanner specifically. It's a 0.064 Tesla scanner. It's a portable MRI scanner, much cheaper than standard high field MRI, doesn't require all of the infrastructure. So doesn't require a dedicated room, for example. It can be moved around, it can be plugged in the wall. And so it essentially, it has several advantages. One, the cost, of course, of maintenance and purchase maintenance and all of those things, which can be a really big factor in lower resource settings. But also it can be moved around. It can be moved towards the patient's bedsides, as opposed to current, current conventional scanners where you have to move the patient to the scanner. It doesn't require dedicated rooms, and so it has a lot of advantages both for clinical and neuro and for research environments. But of course that means also that the image quality is a little bit lower than, or a lot lower than in a 3T scanner, although they've been improving it as much as possible in the last few years. And so these are the different sites for Unity. You can see here all the blue sites in particular are sites where a lot of the data is being acquired for the project. And I'm going to talk particularly about the 3 sites in red here. This is University of Cape Town in South Africa, Makerere University in Uganda, and Aga Khan University in Pakistan. And these are 3 sites where the data for the LISA challenge has been gathered. The University of Cape Town study is led by Kirsty Donald, and the idea is to look at the first 1,000 days of life and trying to understand environmental factors that determine how the brain develops in those in that age range. And they have a ton of data, really high quality data that they're using to do that, and a pretty large database now. And then Aga Khan University, there's a MIND project where again they're looking at early ages, is 1 month to 4 years old, looking MRI and other neurodevelopmental assessments. And they're also using both high and low field data. And the data that were used for LISA, especially for the segmentation task, which I'll present shortly, the data is both high and low field. So both the hyperfine scanner that I showed you earlier and a conventional T scanner to acquire the data. And the idea is to look again at modifiable environmental factors that can affect brain structure and function and so on, and to ensure that children are doing as well as they possibly can. This slide is a tiny bit outdated, but just to show you that these are all the different things so don't look at the numbers of participants, but just the different demographics that they use. So study participants, demographics, they get a whole bunch of demographics, a whole bunch of data on mothers. They look at neurodevelopment, biological samples, and so on. And then finally, Makerere University, again, a large data set of a cohort from birth to 5 years old, and they look at different types of assessments as well, neurocognitive assessments over time, and look at different prenatal and postnatal factors that affect development. And that one is, Makerere University in Uganda. And these are all the different factors that they look at. So these are data contributors for LISA. So just different challenges that we see both. So LISA combines, the reason why we have this challenge is that it combines two of the really challenging things in MRI. So first of all, pediatrics is generally already harder to analyze. Pediatric data is already harder to analyze than adult data for several reasons. One, the contrasts are lower on the images, especially at earlier ages, where the brain has not yet myelinated fully. They're infants, if you don't sedate them, and we don't for this study, this is all normal, healthy infants. I should have said that earlier. So there can be a lot of motion. Traditional tools that are typically meant for adults, and so then adapting them to infants is not always an obvious thing to do. And then this is compounded by challenges from low field MRI, where as I already said, the image quality is also lower. So and so then we have to, and also most methods are made for high field. So LISA was made to remedy these problems. And just to give you a quick example, also, this is what a pediatric high field MRI image looks like, just to give you a sense of what we're up against. You can see here, for instance, the 1-month-old, the contrast is very low. This is what I was talking about, compared to, for instance, a 24-month-old. And there's also, of course, a lot of other changes that happen in the brain during that period of time. So any training set has to have a lot more data to cover all of those different changes in different ages. And then just to show you what ultra low field looks like compared to high field, I'm using interchangeably low field and ultra low field. I just assume that they're the same for this conversation. But you can see the ultra low field image clearly has less definition than the high field image. And this is why we have this challenge, is because methods that work for high field don't necessarily work for ultra low field. And images are acquired typically in 3 different acquisition directions, so axial, sagittal, and coronal. So then we usually combine them for our analyses, but you'll get all of the different planes for the LISA challenge right, and other pre-processing steps that we do on the image. I'm going to skip the details of the pre-processing step just in the interest of time. We also just the one step I will talk about is for segmentation. So we have created a set of ground truths for the segmentation task of the challenge, which I'll talk about a little bit later. But we have basically created a set of manually traced out images, both on high field and low field, that are going to be used, one, to train the high field ones are used to train the algorithms, and actually both of them are used to train, but then also the low field ones will then be used for testing. And here's an example segmentations look like, our manual segmentations. Okay, so I'm going to start talking. I'm going to skip that one, but I'm going to start talking about the quality assurance task. So these are some of the different artifacts that we see in the images that we have. So motion, of course, which most of you will have seen if you've worked on MRI. Distortion, banding, position is a big one in that scanner. We're putting babies in the scanners, and sometimes part of the head is missing, and then zipper artifact, which is a little bit more specific to hyperfine scanners, and noise and so on. So all these different artifacts are issues for our images. So this is what we're trying to solve, and then in task 1. So task one, so to give you, to just quickly go over the tasks for the challenge, task 1A is a quality assurance task. So trying to detect how good the images are. I'll go in the details on that a little bit after this. But and then task 2 is both artifact, or task 1B that is artifact removal and then quality improvement of the images. And then task 2 is a multi-structure segmentation. So all of these different structures here manually segmented. We want to get an automatic segmentation for them. And quick statistics on the LISA challenge. This is last year's participation number. We had 167 participants plus 94 participants from the RISE summer school. And so we ended up having some very, very good winners for each of the tasks. Very, very impressive tools. These are the people organizing. Feel free to reach out to us. And I meant to put a QR code, and I apparently didn't. But do go online. If you go on the MICAI website, you will see the QR code for the challenge. I will say we're currently in the validation phase. You can still join. Feel free to join anytime. Well, ideally as soon as possible because we are in the validation phase, but there is still an opportunity to join the challenge. If you have any questions, please contact me, and I will find the QR code and add it as we have the discussion. Thank you.
Thank you so much, Professor Natasha. I would like to ask if there are questions, 1 or 2 questions for her before we move to the next phase of the information session. Okay, just to clarify, the travel award has been confirmed to be July 1st. If you're able to submit your workshop paper between now and July 1st, you'll be considered for a travel award. And if you didn't submit before then it means you only be considered for just participation in the workshop. Thank you so much once again, Professor Natasha, and I really appreciate the fact that LISA is strictly focused towards underserved communities such as ourselves, and the development of AI methods that will be developed on this challenge will complement all your efforts on the Unity projects in making the clinical application of all the efforts to actually get to the patient eventually. Thank you so much once again. So now that we've heard from Professor Cott and also Professor Lepol, one question that is on the table is how do we exactly submit this manuscript after we've gotten it? And that is what I will be walking us through in a short while. We know for if, okay, just give me a moment while I share my screen. Sorry, slideshow. Okay. So we know for a fact that we have MIRASOL RISE collaboration as part of our effort to building and sustaining efficient technologies for medical imaging in resource-constrained settings. That's the main reason why we are partnering with LISA Challenge to ensure that the two of us can have an handshake and actually get all that is necessary to make clinical impact on our patients. But participating in the workshop alone is not just the way to go. We really feel like it is important we explain the processes that are necessary before you could be able to submit your workshop. The link as shown, mirasol.risemikai.org, is where your application, where you find all the information about the workshop, and there is this specific app on as part of the tabs we have, which is on how to submit. If you click on that, it's going to show you a submission portal, which you could also access using the link that has provided. But specifically, there is a button that you have to click on, which is on the top right corner of that particular platform, which if you click on it, it would lead you to the open review portal. For most people who have been part of Maker challenges and workshop before now, we typically make use of CMT from Microsoft, but there's been an agitation and a lot of push towards making everything completely open source, using open philosophy for everything we do on the international side. And that's one of the reasons why MakerBoard decided to go through open review this time around. If you click on the open review portal, it will bring you to the home page of the challenge itself. I am working on the understanding that majority of us might not be familiar with open review yet. It is an open source platform that gives you the opportunity of submitting your manuscript to um, multiple conferences and workshops. In the same instance, it also allows you to be able to conduct review on the same platform as well. So I would like to employ each and every one of us when the call for reviewers starts going around, let us sign up to review the various manuscripts for the workshop as well. So after you get your home page, you'll be required to log in first. If you don't have an account, you'll be required to create an account in that instance. If you are new, if you have an account already, just provide your email and your password and off you go. And if you do not, you'll be required to sign up. Sign up typically requires just your full name, your email address, information about your facility, your institution, your conflict of interest. And as you provide all those information, it is going to ask you to also verify your email through a link that will be sent to your email. And after you complete that process, you are now supposed to come back to properly now register for the workshop, because in the process of signing up, there are so many cookies that can get deleted, caches that can get refreshed. So you go back to the initial home page for your Maker challenge, where you click on submit for open review portal again. But the interesting thing this time around is that it will have already signed you into your account, as seen on the top right corner here with the display of my name there, and then you can now click on add Maker 2026 workshop MIRASOL submission. If you click on that, it's going to bring you the platform, the interface with which you'll be required to submit all you are required to do for the workshop. And starting on the list, as Professor Mehmet Scott has told us already, we are required to submit a 10-page long paper, 8 pages of content, and in this particular format: introduction, previous work, methodology, resource discussion, and impact on resource constraint settings. After filling that, you type in your title, you type in your co-authors, and as you keep typing them, for some of your co-authors who are already on the platform already, you can just click on the add button here after you search their name. And for those ones that are not on the platform, including Mr. Professor, no, I think it was a Mr. Not really sure. Albert Einstein, is shocking to know that he's not on open review. So if you have people like that, you'll be required to manually provide their information with just providing only the full name and then the email address, and you can add them and they get inserted as parts of your authors for your paper. And you can use these arrows we have here to arrange the order of authors you intend to have, from first authors to last authors, and back and forth. There and then you'll be required to provide a keyword. The keyword is just like a summary of the search terms that your manuscript is talking about, and you separate all these keywords by just a comma. It's meant to be a comma separated value. There and then you present your TLDR, which is like a summary, like one sentence summary describing your paper. You put that in the TLDR box. Then you provide your abstracts, which is expected to be less than 200 words as recommended by Professor Mehmet Scott. There and then you upload your PDF, which should have been in the structure that have earlier been provided. You definitely have to be a PDF. There is no provision for uploading other formats. The licenses, the readers, and everything will have already been configured for you. And at the conclusion of that, you are meant to click on submit, and there you complete your submission process. By this, you are supposed to receive a confirmatory email in your inbox confirming that your submission has been received. In the same instance as well, if you want to confirm if it has been received, you could go back to your home page for the workshop, and if you check your recent activity tab, you would be able to say that it has already been added to the workshop. So once again, I implore each and every one of us to sign up for reviewing multiple papers for the workshop, and also to remind us about the important days as well. 15th July is the deadline for the submission to the workshop, but if you want to be considered for travel award, we are required to submit your papers between now and the 1st of July. Thank you so much on behalf of my team. I would like to take questions at this moment. So we have a question from Zubair Abdulrazak.
Yes, thank you, Tomaruf. It's a nice presentation. I pasted the question in the comment. Please, how do you register AutoShift if it is joint first AutoShift in the platform?
Okay, so we currently do not have provisioning for that for the short paper page, but when we get to the camera ready page, we would be able to make provisions for that.
Okay, thank you.
Do we have any other question? Okay, then I have another question here asking about compute resource grant. Yes, we also have availability for people who want to make some computational work but do not have access to compute space. There's a link on the website, on the MIRASOL website, where you could apply for compute resources. And if you've not received your computational access yet, so kindly send a follow-up email to info.camera.mri@gmail.com, which I'll also be providing in the chat box as well. So do we have another question? It seems not. All right. In the absence of further questions, I would like to appreciate each and every one of us for making it a day with us on this conversation today. I really look forward to reading your manuscripts, and looking forward to you also presenting your manuscripts at both MIRASOL workshop and also participating in the LISA challenge as well. I really look forward to building a larger, stronger, and bigger ecosystem of medical imaging researchers on the continent. And if there's anything you feel we could leverage on each other's support for consultation, for computer science, and also for guidance, please do not hesitate to reach out to the info.camera.mri@gmail.com email address. But shortly before we call it a day, I can see Kairi Ibrahim's hand raised up. We'd like to quickly take your question. Over to you, Kairi.
Thank you, everyone. I do not have an institutional email. I registered on Oppo Review with my personal email, and then they asked me to get an institutional email. And in my institution as a student, they don't provide you with an institution. You use your personal email for everything. So how do I do about that?
So I do not really understand why it will not take your personal email account. I have two accounts on Open Review, both personal, signed up personally with my Gmail, and another one with my institutional email. I would recommend that you try to find out what exactly is the challenge, why it is not taking your email yet, but I expect it to do so.
All right, thank you. I will reach out to you when I try it again.
All right, all right.
Yeah, and I will say in general, if you guys have questions, Maruf, if you don't mind sharing our emails with everyone so that if you have questions beyond that, I put the link to the Lisa challenge in the chat. But if you don't mind sharing that also with everyone,
Yes.
Maruf.
Yes, I'll do that soon.
Yeah, and then thank you, and then feel free to reach out anytime if you have any questions. There's also on the Lisa website, there's an email there like info@lisa something or other. So just feel free to email that link if you're not sure. And if you have questions about also just generally like we do a lot of global health work and on MRI, if you're interested in any of that, feel free to reach out for that as well. We're always happy to hear from you.
Right. Thank you so much. Would you like to give a company closing remark to Professor Mehmet?
Yes, as I said, Maruf, thank you for organizing this and thank you for inviting me. And please feel free to, I believe Maruf will send out my email address. Feel free to reach out with any questions about the paper submission, or if you like what we do, so you can go to our website, kurtlab.com. We do a lot of neuroimaging work. If you're interested in working with us, please send me an email and I'm more than happy to talk with you. So thank you.
Thank you very much. Thank you so much once again for making it day with us. See you next time on our Compass edition. Have a great day. Bye-bye.